animals, what they will do. There is a set time for birds to lay their eggs. There is a set time for predators to come and take those eggs. So all this chain, if it is broken, just see what the havoc is being created, and because of that there is a, I think I was reading somewhere 50,000 species are going extinct every year because of this. We are not, and we are using out of 3 million plant species, we are using only 30 plant species for our food. We are not using anything at all. So the question is, there are a lot of varieties, there is a lot of genetic base, but what we are doing as a breeders, what we are doing is, we are using only 3 or 4 varieties. In case of rice, you will see there are mega varieties. You call them mega varieties. They are grown throughout India. So because of that, what happens? The genetic base, it gets very smaller, because you are using only 2 or 3 varieties, and then you are taking gene from somebody, putting in there. Again, the genetic base, because of your varieties which are hybrids, which we are using. Hybrids decrease the genetic base. They do not broaden the genetic. And what happens is, because as you are students of agriculture, you know that when there is a monoculture or there is genetic base goes very small, what happens is, because of that the plants and that plants, they become very much susceptible to insect pests and diseases. What we need to broaden the genetic base. So these gene banks, wherever the gene banks are, they are the reservoirs of a lot of information. Like when we go to the, suppose I go for these explorations, when we go to the farmers, they have some plants of one variety. They are not using it because they are getting something very good. Like in tomato, they have a landrace, they will not use it because another tomato variety which is a hybrid, they are interested in getting more and more of yield. They are interested in getting more and more of plants or varieties which are acceptable to the clients or the customers. So what they will do is, they will just leave it. And by the time, after 10, 20 years, those varieties, suppose those people who are from villages, they know that there were some varieties which were grown earlier, but they are not there. Now we just lament that earlier food used to have very good taste. Now the food does not have any taste at all. If you go to the supermarket, you will find everything is very huge. Pear, they are such a big. Even your, what do you say, pear or apples or anything you take, they are very big in size. So what has happened is that because of the increase in the quantity, we are losing the quality. About 7.2 million accessions are there in our 1300. You must have heard about the Svalbard gene bank, which is a doomsday gene bank they have kept. So, but these extensions are not used optimally. So what happens, like we have our NBPs here also. There are, I will show you how many accessions are there. The people there, they are working on only, like I have some material, I will just send them, they will just keep it in their gene bank. Then from somebody will bring from abroad, they will take that and they will keep it in gene bank. Their main work is to quarantine that material and to give it to us. Similarly, we have some material, we will just give it to them. But the thing is it a dynamic work? I don't know, because you are not scientists. When you ask them to give them material, they will not give you anything at all. Why? Because they are not taking it out. They are not evaluating it. They are not characterizing it according to the traits. Because why? You need land, you need space, you need money, you need labor. You don't have anything at all. So what they are doing, it has become basically a dump yard. And what happens, suppose in case of, I work on mostly on onion and garlic. In case of onion, the viability is at least 6 or 7 months. After 7 months, if you take your seed and you are happy that you are going to start your experiment, it will not germinate at all. Then there are cruciferous, they have 3 or 4 years. You cannot treat both of them same. So what happens is that because of lack of money, lack of resources, you cannot do anything with it. You don't know what is there. Maybe there is something gold hidden in those germplasm, but you cannot do anything at all because you have not evaluated them. So this is what he is saying, that there is insufficient evaluation of the collections, limited input by breeders during documentation. Suppose I am sending something. I have brought it from some farmer also. We send that material also, but I don't know anything about it. I have just written it is from farmer. This is the, we never used to write latitude and longitude also. Now people have started writing latitude and longitude of that place, and we just send it. We don't know what is the importance of that material because we are not also evaluating. We are just sending it. They have their own people who go for expeditions. for different crops. One person is going for onion, garlic, tomato, potato, brinjal, this and that. He cannot go for anything else. No? So he will just bring it. So there is not any information. So accessions with limited environmental ability, accessible material not always suited to agronomic needs. There are some material which you cannot grow everywhere. As we know in agriculture, one variety cannot be grown here. Like I can, since I work on onion and garlic, I can tell you that there are some varieties which are very good in Maharashtra. When we grow them here, you can get good bulbs, but when you go for seed production of those, they will not grow very well. Then comes your, whether it is a long day, whether it is a short day, whether it is a intermediate day. Since you are from MSc or PhD students, there is one variety, brown Spanish. You must have heard brown Spanish because we also read about the brown Spanish. You bring that brown Spanish here and grow it, it will not grow. You will get the leaves, but no bulbs at all. We get many farmers who come to us that about garlic in Maharashtra and other here also. Why can't we grow Chinese garlic? And it is very difficult to make them understand that why we cannot grow Chinese garlic. Even we had scientists who used to just scold me that why can't we grow Chinese garlic, because it has very good yield. It is very big in size. Most of the people prefer to take Chinese garlic because it is having 3 or 4 cloves. Weight is like 25, 30, 40 grams. Why we cannot grow that? The problem is when you grow it, you will get the leaves, but no bulbs at all. So that you should know. There are some people who will just tell farmers, okay, you grow it, but when they grow, there is no bulb at all. Then what will happen? Farmers will see, and farmers do that also. So adequate quantity of seeds, sometimes what happens that you do not get the seeds you need because they have very less seeds. They will give you the material, they will tell you, you first propagate it, get the seeds, but then it is again a problem. Problem comes where? If it is a self-pollinated, it is simple. If it is a often cross-pollinated, a bit simple. When it is highly cross-pollinated, it needs a lot of money. For one candle, for one material, it needs 50 or 50 rupees, and then you have thousands of material too. Then when you get the seeds, threshing them, crushing them, and then keeping them, it again becomes a problem. So conservation of these is a big problem. People can bring and give. Evaluate that. Pre-breeding aims at introgression of the genes, and then base broadening. We are broadening the base. In case of pre-breeding, we need a close collaboration between the gene bank managers and breeders. Greater likelihood of more complex hybridization issues, linkage drags. It is normal that when we are making a cross between a wild species with a cultivated one, there will be a linkage drag because most of the negative things will come with the wild species, which we do not know because they are wild. That is why they are known as wild species. And raw material for breeding. Don't think that if you are working on pre-breeding, you will get a variety. Every breeder who is working on breeding, he wants varieties on his name very fast. That in 2 years he will have variety, then again he has a number scorecard and he will become principal scientist. So pre-breeding everyone cannot do it. Maybe it happens sometimes that when you retire, you will make a pre-breeding material for the other generation. But we as Indians, we are not interested in giving credit to anyone else. We want to take credit for ourselves only. So breeding results in a new variety. So that is the reason most of the people don't want to work on pre-breeding. I will show you why. So pre-breeding, rule of thumb, it is a multidisciplinary approach. You need gene bank managers, you need plant breeders, and they will make a strategy to combine the resources. How we are going? First of all, the thing is, you should know what for are you going to work. It is not that when you have done your work and ultimately at the last stage you will come to know that whatever we have done for last 15 years, it is of no use. So you should know for what you are with. For that, you should know your crop, you should know your traits, you should know the genes which are responsible for that. If it is a single gene, enjoy, do that work. If it is a multi-gene, bring the money. You need a lot of money. Generating intermediate material, the interface includes evaluation of traits of interest, that is what I said, data management, study of reproductive biology. Whether it is suppose a crop like tomato, you can take 3 or 4 crops in a year, you have to do selfing. In 4 years, if you take 6 generations of inbreeding, 6 generations of selfing, you will get material in 1 year or 2 years. Crop onion, where in 1 year you will have bulbs, in 2nd year you will have seeds. So multiplied by 6 generations of selfing, how many years? So in 12 years you will have inbred material. Couple it with the traits of onion, it is having high inbreeding depression. So when you do first years of inbreeding, from elephant they will become like a mouse. 2nd year you will not get them at all. So you have to see the crop also. And then collaboration hybridization. You cannot do hybridization at one place. You should have different places. In case of people like abroad, they have phytotrons, or they have different methods where they can do it there. We do not have that. So we have to see whether I can make crossing here, whether I can do crossing in Himachal, or whether I should go to South India for crossing at different times, so that I am able to increase my material at a very fast rate. So judicious pre-breeding is no pre-breeding needed. This is very much important, is that we do not need pre-breeding. It is not like a fashionable term that I am working on pre-breeding, I should work on pre-breeding. Why? First thing is, if you have commercially available, adapted and acceptable varieties, you don't need any pre-breeding at all. Advanced selections well adapted to target environment, or gene bank accessions that are well adapted to target environment, you don't need pre-breeding at all. Pre-breeding is needed only when you have exhausted all the known sources of your material for your resistance or any other thing. Then you should know that which are the closely related species which can be crossed with your material, and wild species less closely related. These are, first you have to take the land resource, anything which you have that they can be used. If you do not have, then go for wild species. Then there is a concept of gene pool 1, gene pool 2 and gene pool 3. In case of gene pool 1, when you make a cross, it is very simple, and F1s will be this you are fertile. Gene pool 2, a bit difficult, you can do it, but they will be sterile. In third, leave it. Run away from gene pool 3. So this I got from somewhere, that these are the international gene banks and information network for main crops. I have taken from Trends in Plant Science. If you see here, there are International Rice Research Institute, Biodiversity International, CIAT, you must have heard about all those things, CIMIT, SIP, ICAR-IARI, ICRISAT, IITA, WARDA, AVRDC. We need this if people are from vegetable science. Global Gateway for Genetic Resources Genesis, GRIN, ECPGR. Where is NBPGR? Here. So see, they don't know NBPGR is there. They are writing about only these. Why is this? Is it because they are biased towards NBPGR? When you go to this, I have gone to this Green Global, germplasm, work on onion. You will see what is the material there, and then you will also see, when you click the material, the traits of that material. You will see each and everything in that. In case of NBPGR, I don't know whether you are able to see anything. So they are not able to get anything from there. So only the germplasm banks which are dynamic in nature. Dynamic means they are living, breathing. Every day some material is coming, some material is going. They will tell you which material you can take, and it is free. Taking free. Normally whatever is free, whether it is long day, short day, you don't. So these materials like AVRDC charges material, but the material is going to be that material. When we are preparing this lecture for you, I saw that they have written a paper, what is the impact of AVRDC on international vegetable industry. They just bring the breeders there, and they have 2 or 3 days program in AVRDC Taiwan. They bring the breeders and show them the material in the field of tomato, and other brinjal. They show the material that this is resistant and it is resistant in the field. So then they will choose material from there, that this material, this material, this material, we need that material. Sell it. That is how they earn the money, and then they use that for banks. So in case of this, I have taken from NBPGR. Just see how much material we have. In tomato and wild species 2,557, brinjal and wild species 4793, chili and wild species 4544, ash gourd, bitter gourd, bottle gourd, sponge gourd, ridge gourd, cucumber, muskmelon, watermelon. So this is the latest data from their NBPGR gene bank. They have this material. It is there, carrot 108, radish and fenugreek, and we have a lot of material because they go every year for these explorations. They have this material, and you can just talk to them, and if you are working, suppose whosoever working on these things, you can just try to get some material. So why we want to work on this pre-breeding is because we have some knowledge about disease resistance, pest resistance from the vegetables. We cannot just start working from the base. Now we know that in case of disease resistance, Solanum integrifolium and torworm from brinjal, they are resistant to bacterial wilt. Indicum integrifolium, sesembrifolium, they are resistant to fusarium wilt. Solanum gilo integrifolium, Pomopsis this fruit rot. In case of brassica, there are some which are resistant to black rot. Solanum demisum, it is resistant to potato, late blight, potato virus Y, Solanum chacoense, fusarium wilt, Lycopersicum pimpinellifolium, leaf curl virus, Lycopersicum hirsutum, farmer species grave. That means we know that there are some species which are resistant, and it is based upon the work done by other scientists. Similarly, in case of watermelon mosaic virus, Cucumis metuliferus, tomato mosaic virus, Peruvianum, leaf spot lactica salina, and downy mildew in Allium royale. I will tell you about the story of Allium royale after some time. In case of onion, there is purple blotch, and that was in our time also, that is in your time also. But have you seen any source of resistance to them? No source of resistance. And nobody is working because these are the India. India is number 1 in area and number 2 in production of onion and garlic. Similarly, in case of insect resistance, we have in brinjal, fruit and shoot borer is one of the worst problems. So we have the Solanum ethiopium, Macrocarpum and Khasianum, Colorado potato beetle, Solanum chacoense in potato, aphids, Solanum number 30 in potato, fruit borer, whitefly, nematode and other things. See, we have so much of resistant sources present in case of our wild species. In case of stress, nowadays stress is, you see most of the people are working on stress, biotic stress and abiotic stress, and abiotic stress is salinity. If you see Delhi area or Haryana area, because of lot of this rice wheat system, the salinity has increased a lot, so people want varieties of salinity, or you can for drought also. It is going to be a worse problem. Drought is going to be worse because we do not have water. More crops per drop. So in case of stress resistance, we have Lycopersicum esculentum. In case of vegetables, most of the people you are from, which is the king of vegetables as per tomato is awesome crop. Potato. But in case of the money which India gets from vegetables, which is not, but where is the work? Why? That you have to answer me afterwards. So stress resistance, salt tolerant is Lycopersicum chismani, culinary, flood. So suppose if somebody of you is going to work on salt tolerance, you can take these varieties and then try to make some crosses. But before that, you have to see the literature, whether somebody has done this work. It is not that you will start and afterwards you come to know that it is difficult to make the crosses. I will tell you an example about this Solanaceae or tomato. If somebody, when you start working on tissue culture in tomato, you just take any leaf and throw it into your medium, MS medium, it will grow. Take internodes, they will grow. But anthers, no. Nobody works on anther because it does not respond. There is a patent on anther. So you have to know your crop a lot before you start your work on anything. So after that, we have one project. I am a PI of one project here in pre-breeding. So I just wanted to show that what we are doing here in case of pre-breeding. This is in case of capsicum frutescens and capsicum chinense. We are looking for the resistance to curl disease under field conditions. And we have found that this EC, A787146, is resistant. So that means we are going to use this variety for our cropping program. Then what we are doing is that sometimes when you get the wild species, the problem is we do not have taxonomists. Taxonomists will tell you which variety is, which wild species is this, which wild species is this. But when you do not have taxonomists, what you can do is you can go for DNA barcoding. We are working on DNA barcoding also, like ITS, internal transcribed spaces. Then there are many different barcodes which are being used, RBCL or MATK or TRNH, PSBA. So this is, in case of capsicum, they have used this ITS2, and they have been able to differentiate the different wild species. So NCBI, you blast the sequence and then you try to see how much is the similarity between your sequence. These are the sequences. How much is the sequence similarity between this sequence and yours, and this species. It is just a first. It is not 100% full proof, but it is a estimate. In case of cauliflower, when you talk about what do you say? Making different cytoplasms for male sterile, because for example, where they use cytoplasm susceptible to one disease, so we want to diversify the cytoplasm. In case of cauliflower, we are working on introduction of Diplotex catholica and Trichostoma bali in this cauliflower to make male sterile cytoplasm. So what happens is that when you make a cross, it is not able to survive. So they will do is embryo-rescue. So that is why when you work on pre-breeding, you should know about your crop, you should know about tissue culture. Then you should know about the species also, and then you should be able to do the genomics also in those. Now, the topic is on genomics. Why genomics is important now? First, when breeding they used to do, they will just and then see what are the characters, and on the list they will say that whether it has crossed or not, whether it is a hybrid or not a hybrid. In case of cucumber, we have very rich keratin rich cucumber germplasm. If you see here, Pusa Uday is 1.09 total keratin and 1.06, here 6.58, 9.2, 8.7, 7.9, and here if it is ripe 24. So it is a source of your protein, and if you get keratin from natural sources, it is better than taking those pills for your keratin. Now the next decade is for food as a medicine. Now when you will go to farmer, your doctor, he will not give you medicine, he will after some time he will tell you one cucumber, one carrot, and then your everything is okay. In case of Cucumis hardwicki, we have trying tomato leaf Delhi viruses, and here if you see here, we have tried to see whether it is resistant to this TOLCNDV, and then we are using the specific markers. So this shows us that if it is not here, that means they are susceptible. This is resistant. So your work, it gets reduced. You do not have to go into the field and lay down the trial and then see whether it is resistant or not. So you are decreasing your land, space, labor, but money less as compared to what is there. Now the problem, what is coming in agriculture community is that you have people, students like you or scientists like us, who are very well comfortable to work in labs. And in field, they don't know how to take phenotyping also. Phenotyping is becoming a problem now. Genotyping everyone is doing. Get any student, he wants to work on, sir, molecular marker pe kaam karunga. If you tell him the field, nahi sir, field mein nahi. But agriculture is in the field only. Another thing is that if you don't go to the field and you do anything here in your lab, both the things, they do not correlate. That's the Then this is about alien species. I got some when I was giving lecture like you to one class in Katrai. So one lady said that she has some different species in northeast. So we had just some discussion, and then she sent me the plant. She wanted me to know that what is this. I simply told her that it is Allium sepavar aggregatum, because multiplier onion is mostly grown there. This species is not like that. She gave something new name, and I told her to send me the material, but she sent me the material. It was having small these bulbs like this, and I was very much confident that it may be sepavar aggregatum. It has been year we have not been able to understand what is this material. We are just trying to take the leaves, the flowers, and other things. It is growing, flower is there, and again the flower is sterile. When it flowered, I thought that okay, I will just make the cross, and then I made the crosses. There was very good embryo, and after 2 or 3 days, there was nothing at all. That means it is a cross between sepa and malt. Maybe, we don't know. So you need a proper taxonomist to work on this thing. What we did was we did the DNA barcoding, ITS and other things. And we found this northeast is here. It is showing itself different between Allium fistulosum, Altaicum, Sepa, Cornutum and other things, but it is different here on the basis of one. Another one showed that it is between Allium royale and multiplier onion. So you are not clean, you are not clear about, because ITS you have to use nuclear also, you have to use cytoplasm. Cytoplasm, on the basis of cytoplasm markers which we have used, it is showing that it is from Allium fistulosum. Fistulosum is one other species. The mother is fistulosum, but who is the father? We are not able to. So these are the things, and this material, if somebody is from northeast, you will be surprised. It is only one village which is using this material, and literally you will see mountains full of this material. Total mountains are full of this material only, and they are earning in lakhs with this material, because it grows as a multiplier onion. They will just take the leaves, these plants from the side, and then sell it in the market at 10 rupees for 10 plants, and they are earning a lot. So there is a lot of variability present in India. If we are able to do that, achievement. We just run after others, that there is something, just bring it. But we have a lot of material, and we know that it is showing resistance to Stemphylium blight, because I work on Stemphylium blight, it is showing some resistance to Stemphylium blight. So we can use this material also. In case of bitter gourd, this work is not mine, this work is from different scientists who are working in our department. They are trying to make a cross between this Momordica charantia, Pusa Oshadi. Pusa Oshadi is a one variety which has got very good, it is very good for diabetic people, and this Musa balsamina for different traits, and they have been able to get some plants. Similarly, in case of okra, for this Bindi yellow bone mosaic virus resistance, they are working on these things. And then we come to your favorite part is sequencing. Discovery was done by Watson and Crick in 1953, and see, first sequence of 24 base pair, 24 base, ATGC 24 times, published in 1973. Then came your Sanger sequencing, and then started first generation DNA sequencing. PCR was given by Carey Mullis, and then ABI 310 in 1996. Genome of this nematode was sequenced, second generation, and after that came a lot of machines, 454 sequencing, HiSeq, now Iron Torrent, and then you specify by single molecule, different machines. If you want to go into their chemistry, you will not be able to understand, because everything is made by companies. They just give you a protocol. You have to work according to the protocol. We don't have to use brains here. We have to just follow the protocol. What they have said, take 2 ml, we have to take 2 ml. Take 3 ml, we have to take 3 ml and do it. Then these are Oxford nanopores. See the miniaturization of the sequencers. It is just like a pen drive now. I have seen in NRCB, maybe Kishore Gaika would have shown you. It is just like a pen drive. They are using that. But all these things depend upon how much money you have. If you have a lot of money to use, you are free to use those things. Then data analysis. First, When Sanger sequencing starts, there used to be this much data. This is the whole genome, this is the data. But when you will connect the dots, you will get that was your back sequencing. When you make backs, bacterial artificial chromosome, you will put those sequences and then you will do. It was very easy. It was just like old days, very ambassador, this car, very sturdy, very solid, but less mileage. Then people thought, why don't we go for Maruti? If you go on Maruti, what will happen? Accident, neither the Maruti survives, nor the people who are in Maruti, they will survive. Okay. Then people thought, why not Ferrari? Ferrari is best, but for Ferrari you need money. And when you get a Ferrari, you get so many dots. A person like me who drives Maruti, he will make dots like this. But a person like who is very good, very smart, he can make these dots and make this also. So everything depends upon, this is an art, how you are going to do that. You will get a lot and lot of data in this sequencing, very huge data. You don't even know what to do that, because your computers, they don't work on those. If you think you will do this and then you will have a computer, no. You need workstations. You need full rooms for those things, and on that also it will work for 1 week, 2 weeks, 3 weeks, months. Depends on, because you get a lot of data, how you are going to use that. So you need, then there are many softwares which you are using. It is not a backend, there is a lot of work going on for making these things. And you must have seen, everybody must have shown that the cost of per raw megabase, every time they show that the cost is decreasing. That is good. I am also happy. You are also happy. But who is going to process the data? Where are the bioinformaticians? We have bioinformaticians who are doing MSc and they have done. They will do example of making a molecule of drug designing or last form. But where are the real bioinformaticians? I had one friend from New Zealand, he used to say that whenever I do any postdoc, a lot of applications from India, but nobody knows what they are doing. You need very high quality bioinformaticians. We have a dearth of bioinformaticians, and for a crop like horticulture, we don't have biotechnologists also. So that is why you see most of the world rice, wheat, maize. A bit of potato and tomato, because Rick is working in their USA. So finally, we had this tomato genome in 2012. Have you seen this paper, anyone? Never felt the need to see this paper, good. I also never saw, I saw it yesterday only. So tomato genome published in 2012. We have genome sequences, we have high quality draft genome sequences, then there is the finished. Suppose in case of tomato, they had draft genome sequence. Draft genome sequence means I came to here to visit IARI. I came from one door, went from another door and went back. I just saw what is there. Here are buildings and others. This is draft genome. Simple terms. Then comes your high quality. I came here, I just stopped near every department and I wrote, okay, this is division of vegetable science, this is horticulture, this is this, this is this, this is this. So I know where the departments are. Gold standard means I stopped near vegetable science, I came inside, I saw who are the scientists here, what are they working on, who is the head, and this is the gold, and gold is only in two, Arabidopsis and rice. Whatever sequences you find here, they are only draft genome. Now what they will do, I will show you. This is the site of your Soul Genomics network. If you are involved in this Soul Genomics network, you will see new tomato reference genome. If they have one, why do they need new? So there are again new reference genomes coming every day. It is just like a, in Hindi they say कि भाई कंबल ने हमें पकड़ा नहीं, कंबल ने हमें पकड़ लिया है. You will do one, you have to do more, because when you do first sequencing, there will be a lot of gaps. Then you have to do another to fill those gaps. Then you have to do another to fill those gaps. So you will keep on filling the gaps till you have a 100% clean gold standard reference genome. If you see here, Soul Code, Veritome, they are using varieties now. And eggplant sequencing. Here I wanted to show you completed genome, tomato, potato, pepper, Solanum pennilli, Solanum lycopersicoid, Solanum melangena, and these are Barcoding. These are 150 tomato sequencing project. They are taking 150 varieties and they are doing the resequencing. Here, the cost of resequencing is important because it has reduced. So, whenever you have time, just go to this website, Soul Genomics Network, and just try to fiddle with that. You will be able to understand what is there, that there are markers, which marker do. Suppose I will type drought, just like that, and there will be so many genes will come that they are responsible for genes for drought. You can use those genes. You can see in your varieties as a candidate gene mapping. There are thousands of things which you can do. And then came this. This came first, potato. Potato is very important crop, so we have worked on this. And these are two varieties which they had taken. In one there was long bulbs. If you see these were long tubers and these are good tubers, and these are the chromosomes. Everything is through the your bioinformatics approach. If you see here, everything, and then they wanted to show you the genes, conserve two of them, and what are the genes there and other things. Then they want to see. This is a paper. You can just go through this. And list of sequences, I have just gone through. This is the latest list. Beta vulgaris chenopodiaceae 714 to 758 millibase pair, million base pairs. Amaranthus, they have done brassica, Chinese cabbage. Citrus anatus, cucumis mellow, cucumber, cassava, French lettuce, Solanum lycopersicum, tomato, capsicum, and potato. And if you see here, genome size is latest is 2.5 gigabase pairs. Onion is 16 GB. So nobody will touch that for next 5 years because it depends upon genome size also. You will take only that group which has very less genome size, because when you go for in draft, you have to do at least 5 to 10 times. 10 times you have to just do the sequencing so that you are get. Just yesterday I found that they have done the genomic sequencing of celery also now. Yesterday I was just looking at that. So what I was telling about gene pool is that this is onion or Japanese bunching onion. These are very close to each other. If we go for gene pool 2, gene transfer possible but may be difficult, and there will be some fertility. That is why we can make a cross between Altaicum, Galanthum, Fistulosum. There is a variety delta giant, Beltsville bunching, which is a cross between Sepa and Fistulosum. We are also making cross. The question is that when you make a cross between Sepa and Fistulosum, it will be non-bulbing. There will not be any bulb. That is also a problem of pre-breeding. We cannot use that material anywhere. We need bulb. So when you go for GP2 to GP3, it is very difficult to cross because lethal or completely sterile F1. They will either die, there will be chlorosis, there will be albinos, and they will just die. You will not be able to get. And in that comes your Ampelophrasum, Mekigi, onion, Rachio, and garlic. Garlic doesn't flower at all. There are some reports where it has been shown that the garlic flowers also. So this is in case of onion. Similarly, in whatever crop, you should know what is gene pool 1, gene pool 2, gene pool 3. Then comes a very classical example. In case this is a classical, like I don't know why they don't show this example. Like they show every time the example of sub 1A, submergence tolerance in rice. We don't have any example. They never. It is every time it is rice. Because that downy mildew is a problem in Europe. So what they found was that this Allium royale, this was found to be resistant to your downy mildew. And in 1992, the of downy mildew resistant genes PD1 and PD2 Allium royale, they made a, see after 8 years, they made a genetic linkage map of active population using AFLT markers, and they developed scar of downy mildew. If you see here, segments from Allium royale. They found that if the segments are big or if the segments are even small, homozygous state, something like that, it is not resistant. This is heterozygous. See, there is only one segment. It is very difficult to do this cytology. And Then came one paper, see, I have highlighted it, pre-breeding. Yes, and they are involved in the institute, CGN, Wageningen, which is one of the best institutes for ground breeding. It took them 20 years to develop variety, and I was looking at really developed. Where is that variety? Here, this is the variety, F1. To each and every company, and they are selling it. Downy mildew resistance. And another thing what they have done is patent for that. Funded by private companies, they have a patent for that. And interesting part is it originated from India, and we have never. When I started, I just wrote to a person who had collected this. His name is Eliam Royally. He gave me the address of the village in Uttarakhand where Royally. Then some people, what they have done is that they have transferred the onion to get diversification of cytoplasm. This work is from Japan. They are using, I don't know whether you have seen cytology a bit done, not. How? Similarly, another work that if you make a cross between Sepa and Royally, you will get some embryo and then you take them. Now I have shown that they have done this cytology. These are Eliam Sepa and these are Eliam Royally chromosomes. If you are very good in cytology, only then you can do. But if you are not anything about cytology, so that is where your markers come into effect. Just use those markers. This is Eliam Sepa, this is Eliam Royally, and this is hybrid. Everything is there. You will know that this is a hybrid. It is very simple, na? And then these are, you must have heard about monosomic addition lines. So these lines, there is one chromosome of Eliam Royally, one chromosome, different chromosomes. They have made this Eliam addition line you can use for your pre-breeding program also. So this is just collecting from this. This is from Arunachal Pradesh. This is, people are eating these things. People like this for chutney and other things, and they are very tasty. These are wild mammalians. And these are from Ladakh. They make a cake of these wild species and they keep it, and it is very, they make this cake, they just keep it, and during the winters, and there is very, we don't, enough they will use this for their food. And this is the, this is me here. I didn't go there, he went there. I was not able to go, and so he went here and he got it from there, and I will just take the photos when he will bring. So these are the best photos from Ladakh area only. These are growing there, Persevalanskianum and other things. And these are, we don't even know their names also. This is Eliam Persevalanskianum, and this you have to just. Some examples are that Brisham, I think you will take your lecture here. He worked very good on this Brassica carinata, which was resistant to black rot. And then he found that this, it doesn't have a stem, this tip doesn't anthocyanin. Brassica carinata and F1 is hybrid. Simple to say that you don't need markers also, that it is a F1. He showed that this was susceptible, this was resistant, and F1 was resistant, and then different classes of resistance. These are on the basis of markers, and if you see here, you will find that he just across. He has completed a PhD, and I think we will show you the material also. Material. Another one I like this was that this is a cucumber which is having very high. And they made 2 bulbs. You can do that also. They took this bulb, high keratin. This is low. then they use markers. This is a marker for low, high, and they found that there are some markers segregate with the high keratin content. And out of these markers, they used one marker and they found that this is low keratin, this is high keratin, this is hybrid, and wherever the band is here, high keratin. So what is the important thing is that you don't have to do anything, you just bring your material, you don't have to grow it, take the leaves, use that marker, and then you will come to know, okay, this is high, this is low. And they grow, just leave everything, take those, and on the basis of that, they have used this very big gap, but at least you can use them. Then again, you have to go for fine mapping. For that, you have to use for fine mapping. And this is, you can go for somatic hybridization also. And you have to make somatic hybrids, and then when you get those things, here also the star markers, you can use cap markers to show whether this is hybrid or not. But the question was, how can we be able to understand what is the introgression, how much wild species have introgressed, because that is difficult. For that, there is a paper by, this is the latest paper by Chettilat in Solanum sitiens. They have used, it is a potential source of genes for tolerance to drought, salinity, low temperature stress. There are incompatibility barriers. They made, I showed you monosomic addition lines, they have made introgression lines. The question, they are very lucky genome sequence with them. You have to just sequence these introgression lines, see the reference genome, and then see whether it's come from this and that. What is the part of Solanum sitiens it is in this? What are the SNP markers you can use to get that part? So this is really high tech which they are, and ultimately they have given this type of, they have written, see here, it is plus, this is DF2. Plus means wild type, or you are normal. DF2 means, it is DF2 is somewhere here. I just have to understand where it is. Somewhere, you have to just see it. Where is it, DF2? Every time I just forget, this is DF2. Defoliator, early leaf necrosis starting. So they have given name. That is how you give name to those mutants. So here, style 3, somewhere here, style length. Long style, this is long style, plus is your normal. Similarly, in case of NR3, this is never ripening, they will not ripen, this will ripen. So they have developed a mutant resources. You can use those resources. Even somebody who is working on tomato, you can just write to them and get that material. They send the material, and you can use it for your. So pre-breeding is just like a link between your genetic resources and between your breeding material. So this is what they have done, and ultimately this is a wild species from, see where it is grown. It grows itself in rock. That means it is resistant. But the question is how to use it. We know that it grows in rocky space, that means it is resistant to drought. It will have very high taproot system because onion is a shallow rooted crop. But it will have very good taproot system also. But then it will be resistant to many things also. But how to use that material? That becomes difficult. In genomics, my request to you, if you want to work on genomics, is first thing is that you should have a genome of that. That is important. Then you should have a lot of markers. If you have reference genome, then everything is there. But then you are thinking about the GWAS and other things, you need a reference genome. And then you need, overall, for all those things, you need करवाने के बाद कुछ भी तो बोलोगे, हाँ? You need money. Okay, thank you. Okay.